Welcome to our webinar today, Expanded Panel Genetic Testing, How to Select Your Donor. Everyone who is signed up for this webinar will receive a recording by email that you can listen to later if for some reason you need to drop off. I'm Suzanne Seitz. I'm the genetic counselor here at Fairfax Cryobank. I've been here about 20 years. I've been in genetic counseling about 40 years. And Christy Burke is going to join us as well. She's our Director of Client Services. We will have a very active Q&A session at the end. Our webinar should take about 30 minutes, and then we'll have about 30 minutes of Q&A at the end. And Morgan Barker, our marketing manager, will be moderating the questions. Wanted to introduce you to our genetics team. Cryobank is really in a unique situation. I believe we're the only sperm bank that has a medical director who is a clinical geneticist. Dr. Stern is a clinical geneticist, MD, as well as having a PhD in molecular genetics. So we're really very lucky to have him as our medical director. He's been doing this for decades with us. He reviews donor information. He sets policy for our genetic testing standards, which are always very high. So we do have a very strong leader with our medical director. Brian Mariani has a molecular genetics lab right here with us as well. And so we use his laboratory facilities to do testing on semen for genetic diseases, for genetic markers of infectious disease. So again, very lucky to have that all as part of the cryobank. Our founder originally is also a geneticist, an OBGYN, Dr. Joseph Schulman. And that was his vision initially when he started the Genetics and IVF Institute and then brought in Fairfax Cryobank in the 1980s, was to really have this integrated approach to reproduction and genetics. And Nadine is our genetic assistant. She is part of our client services team, and you may be dealing directly with her too if you have genetics questions. She's a very strong member of our team. So what our goals today are is that we want to talk about what it means to be a carrier, explain how we do our genetic testing here at Fairfax Cryobank, and then with those genetic questions in mind, how you can search our donor search to find a good donor match. And then, of course, we'll have the Q&A at the end where we can answer your specific questions. So we started doing expanded carrier testing back in 2018. That's 6 years ago, more than 6 years ago. The vast majority of the donors on our donor search with available vials have had that testing done. We started with 283 genes that we tested for. We're now up to well over 500. Now it's using the highest detection rate methodology, which is called gene sequencing. That's very important because the genetic testing of 20 years ago was a very different type of testing with a much lower detection rate. So as the years go by, the science definitely progresses and the testing gets more efficient. So when we have a negative result for a specific gene that we test for, it does reduce our risk. It does not eliminate our risk. So none of the testing has 100% detection rate, but it does reduce our risk significantly. You have to have discovered the variant that you're looking for before you can say it's not there. So we do make the assumption there's some unknown variants that have yet to be discovered in all these genes we're testing. But most of these detection rates are well over 99%. So if you were to do expanded panel testing on yourself, and I assume many of you have had it done, the chance of being a carrier is 85%. So you're in good company if you have a positive result. And that's given that you're testing for a few hundred genes. The whole human genome is made up of 20,000 genes. So in a decade or so, when we can test for all of them, we'll all discover that we're carriers for a handful of genes. But right now, there's a good chance that you would get a positive result if you had this testing done. The important thing to remember is that you and the sperm source cannot be a carrier for the same gene, because that's when your pregnancy is at increased risk. That's rare. That happens less than 1% of the time. But those are the situations we're trying to avoid by actively looking for donors that are negative for genes that you might be carriers for. If it turns out that you and the sperm source are carriers for the same gene, now there's a 25% risk for an affected child. This is called autosomal recessive inheritance. And the genes that we're testing for on these big panels are autosomal recessive genes, where carriers are healthy. And it's only when two carriers in the same gene have a child together that you end up with an increased risk for a child with that disease. So this is kind of how the math works out. So two carriers in the same gene, one out of 4 pregnancies would have no abnormal variants. 2 out of 4 or half would be carriers like their biological parents, and 1 would have received the abnormal variant from both the egg and the sperm source. They have nothing there to compensate and make any healthy protein product or whatever that gene is programmed to do, and hence whatever disease that gene results in would occur. So the way we reduce our risks now that we have all this new data that we can have access to, if you are a carrier for a specific gene, we simply need to make sure that the donor has been tested and that he's negative for that exact same gene. Even though there are condition names that sound similar, type 1, type 5, we want to make sure it's the same gene because that is the function, the biological function that we want to make sure is working correctly by testing negative. Now, in converse, if the donor is a carrier for a specific gene, we also want to make sure that you have been tested and are negative before you use that donor. So it goes both ways. Whatever the donor's a carrier for, you're tested for, and whatever you're a carrier for, the donor has been tested negative for. So if you're a carrier, there's a way to look at donors that have already been tested, and we're going to go through a step-by-step process on how to do that. But first, I want to talk about what if you haven't had expanded panel carrier testing done. So at this point, you're just looking for potentially using a donor. You haven't had the testing done. So what are your options as far as looking at donors today? Well, you can look at donors that are not carriers. So if there's no red flag in the donor, then there's nothing to address in you specifically. So we have about 160 donors who are not known carriers. So whatever testing they had done, either on the expanded panel or before, was all negative. Only about 50 of those 160 have had the expanded panel testing. So now you've reduced a list that has almost 400 donors down to 50 that you can look at. So your selection of donors is going to be much less. So obviously the best approach before you choose a donor is to get your expanded panel testing done, know your results, and now you know if you can match with these donors or not. It allows you a much quicker way to make a decision on a donor. But if you do look at the other donors that happen to be carriers and you like one, there is obviously the option for you to go back and get your testing at that point to make sure you address those specific genes that he's a carrier for. So the way you're going to approach this task of finding out what a donor is a carrier for and what he's been tested for is you're going to look at his specific donor page and you're going to download his genetic testing summary, or at least click on the download button and open up that first face sheet where you can see what he's been tested for. It's in a grid pattern. It'll show you the tests he's had completed. This is the form that you can download and share with your clinic as they're trying to help you confirm that this is a good donor match for you. Let me go through what each of the tests are that we do. All of our donors since the 1980s have had a chromosome analysis done. Now, chromosomes are not specific genes. These are the whole structure that hold the genes on them. So within 46 chromosomes, there are about 20,000 genes that are distributed throughout them. But we're looking at the chromosomes because if someone has an abnormal amount of chromosome material, either too much or too little, they tend to not be healthy. So clearly they're not eligible to be donors. But if we have the normal amount, but it's been rearranged such that a piece of one is attached to the bottom of another, you're going to have the right amount, be healthy, but you have the potential of producing gametes, either egg or sperm, that are unbalanced and obviously predisposed to miscarriage or abnormal outcomes. So just as a way to kind of get things started on these donors, we want to make sure their chromosome analysis is normal so that there isn't that increased risk. Typically, this is a test that would be ordered after a couple has had multiple miscarriages to see if this is a reason, but we've done this straight away even before he's had his first reported pregnancy with any of our recipients. We also are doing a hemoglobin evaluation. This is not a genetic test. We're looking at markers on the blood cells to tell us about 3 specific diseases: sickle cell disease, beta thalassemia, and alpha thalassemia. All those tests are also tested by DNA on that expanded panel, but these are just health markers. Is there anemia? Is there infection? You know, these kinds of things are important. We do that for all donors. If it looks like he might be a sickle cell carrier, that's often it will be confirmed on the DNA testing that's done on the donor as well. So the expanded panel testing, we've got quite a few labs where you're going to see varying reports. So that does make things confusing. And in addition, you may have had another lab that had your reporting done. So just as an example, we started with the Sema4 laboratory doing 283 genes. We then upgraded to 502 genes. Then we moved to Invitae to do 514 genes. Now we are doing Natera at 549 genes. We started doing that testing a few months ago, and the first vials for those donors will release soon. So you won't actually see Natera yet. But Natera is used by many clinics throughout the country, so your report may be Natera. But it's important to remember that every lab that lists these genes lists a different panel of genes. So we cannot assume that if the numbers are similar on your panel and the donor's panel, that your gene or his gene are definitely included. So it's always important to check by gene name what's going on between the two reports that you're comparing. Some of the reports list the genes alphabetically. Some of them list them by condition alphabetically. Each report is at least 10 pages long, so it can get kind of confusing. But that's a good reason to get this whole thing downloaded on the donor that you choose and have that available to review with your healthcare provider. So when we're on the donor search, how do we get to all of these different features? So you are going to open up the donor search, find a donor you like. You'll see a little mini box, either a square or a horizontal box, and then you have the ability to click through to the next page, which gives you all of his information. When you see this banner at the top that says added bonus, this donor has expanded genetic testing, you know this donor has been tested for 283 plus genes. And then you can download the genetic report. You can also use our search to help you find a donor that's been tested negative for a gene that you're a carrier for. So you can see that we have a basic search, an advanced search, but there's also a genetic testing search. So the way you use that is you select one of these boxes to dictate how your search is going to proceed. If you choose any, then all the donors are going to be presented. That is going to be close to 400 donors, and even the ones that were tested prior to starting expanded panel testing will be presented. And now that would give you the ability to look at everybody, and especially if you've had a panel done and you were all negative, let's say, then any donor on the list is really available to you. So you could also look at only the expanded panel disease tested donors, and we have close to 300 of those available with vials. You could look at the ones that we tested on the expanded panel that were all negative. And if you recall at the beginning, I told you that that's a pretty small subset of the group, because 85% of people who have the testing done are going to be positive for at least one thing. So now you've reduced your potential list to a much smaller list. But if you are untested yourself, if you have not had the panel testing done, this might be a good place to start looking. And if you find a donor there that you like, it might allow you not to have to be tested right away. Of course, your clinic may want to discuss that with you further. So you can also sort all of our donors by what they were tested negative for. So this is important. If you are positive for a specific gene, you want to be able to make sure the donor you choose is tested negative for it before you even take a look at him, right? You don't want to like a donor that is positive for the same thing you are. So usually when your results are presented, there is a disease name, and in parentheses, there will be a gene name. So you can use the disease name to sort, or you can use the gene name to sort. I prefer to use the gene name because those are consistent across all reports, all laboratories, but the disease name can often have different variations, different, it can be very confusing when you can't find the exact same condition name, but the gene name is consistent, even if the condition name varies. So the way you use that is you open it up, you look for the condition name, and you check it, and you hit apply. And the same thing happens with the genes, which are alphabetical. Again, this is what I prefer to use because I think it's much more it's much more accurate to do it this way, and you're much more likely to find the gene that you need to find. Now, before you hit submit to allow you to look at all the donors that are applicable, let's make the list one that's more useful to you. So you can, in addition to adding the genetic criteria that we just talked about, you can also add ancestry, education, availability of adult photos, what vial type you may need, and whether or not a pregnancy is reported. For example, these tend to be the top things that people use in their selection criteria. But at least that way you put in all the factors that matter to you and then hit apply, and then you can see if you've got a good group of donors to look at. If you've put in too many things, you may have excluded too many donors and there may not be many choices. But certainly look at the genetic search as a way to add to the other criteria that matter to you. So you've now used this search to identify donors that are negative for what you're a carrier for, but we see he's a carrier for something else. So the next step is to truly make sure that you have been tested for that gene and are negative. If you've not been tested, you do need to stop. You either need to keep looking or you need to consider being tested for that gene before using that donor. Now, this is called a suitability matching. If this is something your clinic can do for you, that's terrific. We do have myself and Nadine available to help with that service here, and you can just reach out to us. We would need to see a digital copy of your results, which would then allow us to compare it to your donor, the donors you choose. You can't give us the whole list of donors, but you can give us a few donors that you like, and we can help you. But it might just be easier just to keep looking and see if there's a donor that you like as well, and see if indeed what he's a carrier for, you have been tested negative for. So the way you know a donor is a carrier is under that genetic testing button, you will see something called the acknowledgement of carrier status. That tells you that that donor is a carrier for something. And it also shows you a PDF download of what we call our acknowledgement of carrier status form. That does need to be completed and signed. It can all be done online as a DocuSign before that donor can be purchased by you. I want to review what that form says to you. It will give you the donor number. It will tell you exactly what he's a carrier for, the condition name and the gene name. And then it will suggest that you discuss these results with your healthcare provider before making a choice. It also wants you to acknowledge that if both the egg and the sperm source are carriers for the same gene, that there is indeed that increased risk of 25%, and that additional genetic testing and counseling may be indicated. We do have a refund policy. What we find is that people find a donor they like. He may have limited availability, but for them to go through all these additional steps of testing and getting a suitability match and talking to their clinic, days and potentially weeks can go by. So you do have the ability to purchase those vials. And if we do find out later that you and the donor are a carrier for the same condition, you're eligible for a full refund on those vials as long as they haven't been shipped. They still need to be with our facility. So that gives you the ability to put some vials away on a donor you know you like, takes off that incredible pressure to get an answer quickly, and then you can find out if this donor will work for you. If it turns out he doesn't work for you, we'll do the refund. But we also have the ability for you to keep a vial in storage and exchange with a different donor at some later point. So that option always remains available to you to do an exchange. Whatever you paid for those original vials can be credited towards the new donor vials. But this way you can kind of hold on to those vials of a donor that you really like. We do offer additional testing on the donor. So let's say you've had some carrier testing done. The donor has not been tested for that gene or condition. We do have the option to see if the donor can be tested. Almost all the time, I do have a DNA source on these donors I can use for testing. Results come back usually in about 3 to 5 weeks. There is a fee involved, and we do ask that you own vials of the donor that you want tested before you get started. Since it can take upwards of a month to get results, we don't want that donor to sell out, and here you are, you've already invested all this energy and time and fees to get this testing done. So we do have the ability to test. There are some genes we won't test for, adult onset condition, cancer genes, things like that. So you do need to check with us to make sure that the donor has DNA available and that we can do the testing. That's coordinated by myself and Nadine, and you just call into client services and you can get that started. So perfect scenario here is you have found a donor you like, you are a carrier, the donor has tested negative for that gene, the donor's a carrier, you have tested negative for that gene, both you and the donor are not known carriers. So those are situations that are all green lights for you to be able to move forward. If you're a brand new client and you've never ordered from us before, you will need to complete a terms of use. That can be done before you start your vial purchase with us on the phone, or before you ask for suitability match, or before you ask for any special testing on a donor. Once you've got that terms of use letter completed, you'll have the ability then to move right into purchasing a vial and ordering testing if needed. That's a link here in the presentation. You can use that, but it is a relatively long document and it's worth your time to review that and not feel like you're in a rush to have to try and do it while you're on the phone with client services. We do ask new clients to call us to place that first order. We want to make sure we have everything we need, your address, your clinic's address, the donor, we can discuss donor availability with you, our notification process for a donor that may not have vials available yet, or is getting ready to release additional vials. We can explain the process to you, even help with the donor selection process. So new clients really should give us a call. You can also, if you've done this before and you've ordered vials from us, have your account set up, you then have the luxury to go online and actually order vials online, which allows you to kind of check almost daily if some availability is there and place your order. So just to summarize, it's important for us all to remember that being a genetic carrier is normal. All of us are carriers of something, even if it hasn't been discovered yet. The important thing when we have a child is to make sure that the partner that we use is tested negative for anything we're positive for. That's our conscientious way of reducing risk for our children. And we do have the ability now to use the donor search to help us find a donor that's a good genetic match. And you can always ask us for help to confirm that you are a good match. And then once you find the donor you like, hopefully now you know the steps to move forward to complete your order. We do have a promotion for anyone that participated in this webinar today. If you are still looking and would like access to the donor information at no cost, you can access this free promo code. It's online, and you can use it when you check out, and it will give you the unlimited access package for 3 months. And that's available through the end of October. So now we take it to the Q&A session. If you have any questions, be sure and put those in the box, and we will get to those. Morgan, do we have any yet?
Yes, we just got a couple to come in. The first one here says, if someone pays for an extended testing for an individual and the donor is positive for that gene, will you update the profile and or the offspring?
So if I'm doing special testing on a donor for a specific gene and I find out that he's a carrier for that gene and that was not previously presented, obviously, to the people purchasing, that is something that every single person that purchased that donor vial, not just the ones that have reported pregnancies or births, will be notified of that information. All clinics, in addition, will be sent a letter with the client name and the new donor information. That information will also be posted online in the genetic testing summary. It'll be on that front face sheet, very prominently displayed. Any carrier status is going to be in red, and that information will be put everywhere. So as soon as we get a positive result that we didn't know about before, it will be posted. But we don't do, we only test donors for targeted genes for which your pregnancy would be at risk. So you can't call us and say, please do an expanded panel of 500 genes on this donor because I'd like to have that. We will test for targeted genes for which you are a carrier and your pregnancy would be at risk. So those are the genes that we are willing to test the donor for.
Okay, great. The next one here says, if the screening notes say that I was inconclusive for a gene, should I exclude it on my donor search just to be safe?
So there are some of these unusual genes where you can get these inconclusive results. And probably the safest thing to do, since you can choose from so many donors, is to go ahead and exclude donors that tested positive, you should exclude, but make sure a donor is tested and negative for those conditions before you choose him. If you have any questions, you can reach out to me directly and I can tell you if it's how concerned we need to be, depending on what the gene is. If, for instance, you are an inconclusive carrier for Tay-Sachs based on enzyme studies, which are not genetic studies, that's a different discussion than if we have a genetic result that is ambiguous. But the safest thing is obviously to assume by default that you're a carrier, because that's the worst case scenario, and then exclude donors that have tested negative.
Okay, great. And they did confirm it was Tay-Sachs, so.
Yeah, I figured that that's what it was.
Okay.
So then the DNA testing, just make sure the donor's been tested by DNA for HEXA gene, and then you're good to go.
Great. The next one here says, if you and the donor are positive for the same gene, isn't PGD an option for if you're doing IVF?
Yes, if both egg and sperm source are confirmed to be carriers for a specific gene and it's the same gene, we now know the variant in both of them. Those can be tracked into an embryo. And then you could sort out the embryos that are affected, which would have two abnormal genes, or and then only use the ones that are carriers or completely normal. That is an option. But if, the only scenario I see that happening is if you already have a child by the donor and you really want a full sibling and you're just really determined to use the same donor. But if you can choose a different donor, you haven't started your journey yet, and you can choose a different donor, you're really not hurting anybody's feelings here. You can just go ahead and get back into the list. There are hundreds of donors to look at and find one that doesn't require all that additional effort, which is an IVF cycle, and embryo testing, all of which are incredibly time-consuming and costly, when you can choose a donor that would be low risk and you could do an IUI or do an IVF cycle without doing all that embryo testing.
Okay. And this one says, you mentioned you all do chromosome analysis. Sometimes the donor shapes of the chromosomes are abnormal. Is this reported in their information?
So we do a chromosome analysis on all donors. We report it. Sometimes there are variants that are not considered clinically significant. So there are variations in his chromosomes that have no clinical impact on outcome. So those are reported. It's basically a way of saying normal chromosomes, but we saw something that we see in the normal population that has no consequence in pregnancies, but it could be seen in a fetus when their fetus is being tested, a child resulting from that donor. And we just want people to be aware of it because it would have no clinical significance. It would not cause disease. Now, you mentioned shapes. Now, when I show you the picture of the chromosomes, they are all over the place. They're very squiggly. They don't line up perfectly straight. Those aren't abnormally shaped. Those are just photographed at a certain stage of division, and they just happen to be in that shape, but they're not abnormally shaped. You've already looked to make sure all the light and dark bands are present in the sequence that they should be, which is how those are analyzed. But if a donor has an abnormal chromosome, like that unbalanced arrangement that I was talking about, those donors are excluded and never allowed to be in the program. That's why we do it. We will exclude donors that show these translocations that predispose to abnormal outcomes. Those donors are excluded.
Okay, the next one here said that this question is more related to the CMV status. What is the probability of the baby contracting CMV if one parent is positive and the other is negative?
So CMV is a virus. So if you've been exposed to CMV, you will develop antibodies. After your illness has passed, you will still be tested CMV positive, not infectious, but you'll have positive for antibodies. So if you are CMV positive, you as the prospective mother are CMV positive, you can use any donor, CMV positive or negative, because you've already had the virus, you already have antibodies, and you will not contract CMV again. You have, unlike some of the other viruses that you may have heard of, CMV antibodies last a lifetime. So these antibodies will always be there protecting you. If you are CMV negative, that means you have not been exposed to this virus and developed antibodies, which means you need to be extra careful. The worst case scenario is you are pregnant and you contract CMV during your pregnancy, anytime during your pregnancy, and you become sick and it infects the pregnancy and a baby can be born with many complications. That's the situation you want to avoid. We do test our sperm donors for CMV. So if a donor is tested CMV positive, that means he had a past infection, he has antibodies now. We've confirmed that he is not infectious now because there are multiple ways to test for CMV, and we've confirmed he's not infectious. So he's simply a donor with antibodies. It is exceptionally unlikely that he would cause someone who's CMV negative to become infected with CMV because he's been tested to make sure he's not contagious. But, it's 99.999% chance that he would he's not going to pass on an active CMV infection to someone who uses his semen vials. The semen has to touch you to potentially cause a contagion to pass. IVF, for instance, there is no semen touching you, so often IVF clinics do not have the same rules about CMV positive negative situations because the chance of the infection is almost zero. But if you are doing IUI and you're CMV negative, often your clinics or doctors will say, just to be on the safe side, even though the risk is exceptionally low, just choose a CMV negative donor. That means he also has never been exposed. He's also not contagious with CMV because he's never tested positive. And therefore, that's the safest scenario. We do test our donors every 3 months for CMV by DNA. in the semen and in the blood. So we are really sure that these donors are negative. And of course, the vials are quarantined for a full 6 months before they release. So he's had multiple CMV tests throughout that period of time. So I just think the chance is exceptionally remote, but you do need to follow the guidance of your physician on what kind of CMV status to choose based on if you are CMV negative. Again, if you're CMV positive, you can use any donor. If you're CMV negative, you do need to be listening to what your doctor says.
Okay. The next one here says, are all Afro-descendants donors automatically tested for sickle cell anemia since it's a more prevalent issue in these communities?
You know, that's the way it used to be 20 years ago, where anyone that had African American ancestry was tested for sickle cell. Now, every single donor, regardless of ethnicity, is tested. So we have been doing sickle cell disease testing on all donors for the last 7 years, so regardless of ethnicity. So there are certain diseases that occur more commonly in certain ethnic groups. And it used to be 20 years ago that you would divide the donors into different buckets based on those ethnicities and test them differently. That all went away when we started doing the expanded panel testing. We just test everybody for the same list of diseases, and all those ethnic diseases are included on that list. So that would be sickle cell for African Americans. For Italians, it would be beta thalassemia. Mediterranean population. For an Asian population, we have alpha thalassemia. And the list goes on. There are dozens of diseases.
Okay. And do you do the new extended panel on only new donors, or if a donor is still donating, do you update the profile?
So we test the donor at the screening time. When he first screens, that's when he's tested for the expanded panel. All new donors have been tested for an expanded panel since that date in 2018. And then we have to review those results. And then he is, as well as all the other genetic testing, the screening, the physicals, the infectious disease testing, all of that is reviewed during that screening time. If he is accepted into the program, that those results are posted on his genetic testing tab on his page. If we do additional testing on the donor at a client request, let's say someone wants a specific gene tested because he hadn't been tested for it yet, all that information will be put on his genetic testing summary when those results come back negative. And as I answered in a prior question, if it turns out he's positive, that's posted there as well, and everyone would be notified.
Okay, so the next one here, it says, I'm confused. When you say you don't test for certain genes because it doesn't affect the pregnancy, what if it affects the possibility of the child having that certain disease? Or is it the same thing? What genes do you test for? Could you give me an example?
Yeah, it's a good question. There are some, one example I have is hemochromatosis type 1, the gene HFE. So that is a relatively common gene. About 1 in 6 people carry a variant in that gene. And if you have 2 abnormal variants, then as an adult, you are at increased risk for a condition that is easily treated. So children do not get the condition, and it's considered a treatable condition in adulthood, and not everyone with 2 variants even gets sick. So it's one of those things where we really want to focus on serious pediatric illnesses, and especially those with no treatment. You know, that's the focus here, is to prevent sick children from being born. There are also some cancer genes like BRCA that cause breast cancer. We aren't going to test a donor for an adult onset cancer gene. When you do that kind of testing, it needs to be done in a very conscientious way to respect that he needs to understand the results that would come from that and how it would impact his family. If he has no family history of cancer, just to do that testing on him at a client's request is a big ask. So some of these genes are not done because they don't cause disease in most people, and some of them aren't done because they involve Alzheimer's genes that are predisposing, or Parkinson's genes that predispose that don't actually diagnose the disease, or or cancer genes, for instance. Those are some examples.
Okay. You said that all donors are blood tested for genetics, correct? There was another way for genetic testing that doesn't always show everything, so I just wanted to be sure.
So when the donors have their genetic testing done, we do a blood draw. So there are other ways to get DNA from a donor. You can, or anyone who has this expanded panel testing done, you can do blood, you can do saliva, or you can do a buccal swab, which is a Q-tip in the cheek to get some of your cheek cells. So we do the donor testing by blood. When we do special testing, if the donor doesn't have DNA from blood available, we will use a semen vial that we have reserved. That's not one that you own, where we do have the ability to extract DNA from his semen sample, which is the same DNA that he has in his blood, and we can use that for testing. But when we do the expanded panel testing, we're doing it on blood.
Okay. And then this one says, can you explain more about the genetic testing for purchasers that you offer?
You mean the things that I help people with as a genetic counselor?
That's all they said. For that person, if you want to put in some follow-up information, I'm happy to look at this in a second. We can move along to the next one. In the meantime, when will Natera donors be released?
That's probably days or weeks from now. It's very close. We have several ready to go up, and as soon as they release their first vials, after their 6-month quarantine, they will go up. So but we won't put up 100 at once. We'll put up maybe a dozen a month. So it will be slow going before they make up a big part of our donor list.
Okay. I haven't heard back from that other question yet, and that was the final one. So I think to that person, if you want to follow up with the CS team and get in touch with Suzanne or whatever, I think that would be good. We can kind of follow up with you one-on-one.
Okay. And I am available. You can reach out to client services if you want help with suitability matches. Sometimes it can take a while for your clinic to get back to you and you really want to make a decision on a donor. We can usually turn it around within a day. If you send me your genetic testing results and some donors that you like, I can tell you which ones are good matches and which ones need additional testing either on you or the donor so that you can kind of move forward with a plan. I know lots of people try to get into these reports themselves and it gets very confusing. It's hard to find the gene names and make sure they're the same ones. The reports look different. The results are presented differently. So if you need help with that, Nadine or I are available to do that. We usually do that through email and we send a summary back on the suitability for that. And then we help with special testing as needed.
Okay, great.
So just remember, you will get a copy of the webinar and you can use the promo code if you still need unlimited access to see donor information. And you can use it, it says February, October. So I'm assuming it's through October 27th, 2024. And then you can certainly email us with questions if you don't want to call, and those get forwarded to me as well. So thank you, everybody, and reach out if you have any questions.